Good afternoon. Welcome to the 2026 first of the Biotech Tech Talk series that is starting today. Today, the Biotechnology Center is hosting 10x Genomics, one of the vendors that provides multiple technologies that we have within the Gene Expression Center, ranging from single cell to spatial transcriptomics. Today, we have Egon Ragini here from 10x, who's one of their amazing FASs, who's going to talk about their 10x genomics pipeline, where they're able to take the data from single cell and feed it all the way through spatial transcriptomics. They'll show you how you can combine these data sets and the usefulness of using the multiple software programs that they have available. As I said, this is the first for the 2026 seminar series hosted by Biotech. In February, the Gene Expression Center will also be hosting another seminar. This one would be given by Thermo Scientific, and this speaker will be Doug Gibbons, who's one of their product specialists for their genotyping assays. So that will be next month on the 19th of February. So today, I'd like to welcome Egon, who will give us the seminar, which is entitled A Live Demo of 10x Genomics Data Software. Thank you.
Thank you, Sandra. Thanks. It's great to be back here. Hello, everyone. My name is Egon. I'm one of the science and technology advisor for 10x. I'm here with Benton, your account executive, Eric, your senior specialist. For those of you guys who are on Zoom, Benton is also monitoring questions. So if you have questions, for you guys, of course, if you have questions, just raise your hand and feel free to ask me. What I would like to do with this presentation is, so there's going to be a live demo of the data set, and I have three different, essentially, I'm going to show you how to use three different data sets in kind of an integrated fashion. But before getting to that point, I think I'm going to spend the first 20-ish minutes reviewing the platform that we're going to use for the live demo, if that makes sense. And again, if you have questions, please feel free to ask me, and I'll be also more than happy to share the slide deck if you need it. And so I usually start with this slide because also it's really relevant to what we're gonna show later. And so at 10x, we have this really unique toolkit of platforms that we've been developing to unpuzzle the complexity of biology. Chromium includes everything for single-cell genomics. Visium is our spatial transcriptomics platform. Now it comes with single-cell scale resolution, and then Xenium is in situ analysis. And you guys have all three platforms here. So really lucky to do for that. Now, those three platforms have generated now more than, I believe now we passed 11,000 publications. And this is one of the reasons why we are the single cell and the spatial genomics company you can find in this field. We also have now, when it comes to single cell in particular, we also have the most comprehensive single cell multi-omics portfolio, which means that on top of the typical gene expression analysis, you can profile additional analytes, such as, for instance, you can look at cell surface protein, intracellular protein, regional accessible chromatins, TCR or BCR chronotypes, so on and so forth. That's really important to better understanding the complexity of biology. We have solutions that are compatible with pretty much any species. Even if you're working with bacteria, I would say there is a way around that. So if you're interested in that, just let us know. And then in terms of sample type compatibility, really here is where we have a wide spectrum of samples that can be compatible, including archived FFPE tissue. And I'm going to touch based on that later. Now, those are the three families of single cell solution we have. Not going to focus or not going to talk about universal or AP assay. I'm going to focus on Flex because Flex is one of the data set I'm going to show during the demo. And so Flex is a solution that we launched a couple of years ago. And in a nutshell, what you can do is to take your samples, fix your samples in 4% formaldehyde, and then store your samples at minus 80 degrees for up to 12 months. So essentially blocks or preserve the biology of your sample, really no matter when you collect it or where you collect it from. So it works really well for longitudinal studies, you collect one sample today, one sample next week, one sample next month. But also if you are collecting samples on a Saturday afternoon, you don't need to worry too much about that. Just fix everything and store samples at minus 80. Recently, couple of months ago, we launched a new version of Flex. And so one of the capability of Flex was the ability to multiplex samples together. So running multiple samples in the same reaction, and that helps a lot with reducing the cost per sample. Well, a couple of months ago, we launched Flex V2, which comes with a significant improvement in terms of the number of cells that you can profile per lane of a chip, and in terms of plexing. So when we look at the number of cells that we can profile per lane, now we are up to 1 million. And a full chip will be 8 million cells that are captured in less than 6 minutes. And when it comes to how many samples we can pull together, now with Flex V2, we can go up to 384 samples. You don't need to have 384 samples ready at the same time to run the kit. You don't need to have 200. You don't need to have 50. And that's because the kit is not single use only. So you can use whatever reaction you need, store the kit back at minus 20, and then go back to the kit and use additional reactions. That is really gives you a lot of flexibility. Very briefly, when it comes to the chemistry that we use with Flex, it's not a poly-A-based chemistry. We use probes. Those probes target all protein coding genes in the human or in the mouse transcriptome. So it's human-mouse specific. And we included oligos that essentially this is what allows us to multiplex up to 384 samples together, because essentially we attach unique barcodes during one of the hybridization steps. The RNA target site you see there, that is only 50 nucleotide in length. And so even when the RNA is partially degraded because of the fixation, we can still detect the signal. Plus, the vast majority of genes have at least three target sites. So even if one is compromised, there are 2 more. With Flex, we can also design custom probes. We have a really good tech note that allows you to design a spiking custom probes. For example, let's say you want to detect GFP or a viral transcript in a human sample. We can do that. And again, if you want to do that, just reach out to us and we're going to help you get started with this type of approach. Flex is also a multi-omic solution because we can look at cell surface protein, intracellular protein. We can do both. And we can also run CRISPR-mediated or look at CRISPR-mediated CRISPR-mediated gene perturbation by using custom probes. If you are interested in CRISPR screens, please, please, please come and see us or talk to Sandra as soon as possible, because really we will make sure that you set up your CRISPR screen properly from the get-go, from the very beginning. Now, I also want to briefly mention that Flex is by far the most sensitive single-cell chemistry you can find in the field. And this is not my word, and it's not our marketing department word. There are studies actually showing that. And this is a comparison done by Genentech. And what they show here is, so what you see here is gene sensitivity, assay sensitivity in terms of genes detected at a certain read depth. And the next FRP is what we used to call Flex. Now it's called Flex. But essentially, Flex is by far the most sensitive chemistry. The second most sensitive chemistry in this study was our 3' chemistry, not the current one, the one we launched in 2021. Now the current is essentially on par with Flex. And everyone else is down here below. This is one study, and then there is a second study which actually uses the current version of the 3' chemistry and Flex. And this was done by Fred Hutchinson. And again, it shows that 10x is by far the most sensitive chemistry in the field. And I mentioned that because we hear that other companies say, well, actually, we can generate data which is as good as 10x. Well, that's not true. The other thing we hear is 10x is expensive. Well, it's no longer true. So this is what the Flex kit cost. And the price you see here, it's only 10x reagents, so they don't include sequencing, they don't include core fees. But essentially, if you're looking at, for instance, a 96 sample kit, you're looking at $250 per sample. And with the 384, we are down to 170. So it's not very or much more expensive than bulk RNA sequencing. But think about how much more data you can get with single cell, and think about what you need to generate in terms of data when you're submitting your grant, when you're submitting your paper. And again, you don't need to have 96 or 384 samples ready at the same time. Kits are not single use, so you can use whatever number of reaction you like. With Flex, we also have the ability to profile archived samples. And so what we can do here, and we have a protocol, so you don't need to optimize anything. But essentially what we have here is a protocol that takes scrolls, 10 micrometer scrolls of FFP blocks. We isolate nuclei, and then we profile nuclei with Flex. And the ability to do that is, again, is something unique when it comes also to combine it with spatial transcriptomics. I would say unique to 10x, because now we can take consecutive section of the same FFP block and generate single nuclei data and spatial transcriptomics data, not just from the same type of sample, but from the very same FFP blocks. And there are a couple of examples in the literature already that I'm not showing here in the interest of time, but actually this is one, but again, I'm not going to talk too much about that. That actually takes me to the next part of my talk, which is spatial. Now, single site is great, but there is one piece of information, as we're going to see it later during the demo, that we missed, and that is a spatial context. So we no longer know where cells or genes are expressed across the tissue. But now we can do that with Visium. And so Visium is our platform that allows us to look at the entire transcriptome of a tissue section and at the same time maintain the spatial context of where genes and cells are located across the tissue. We launched an instrument which is called CytAssist, and that allows you to essentially section your tissue on plain glass slide or regular glass slide, stain them with H&E or immunofluorescence, so that you can make sure that what you are sectioned is really what you wanna use, wanna profile. Then there is a hybridization step, and essentially the CytAssist will transfer the analytes from the tissue onto the Visium slide for barcoding and library prep. That also allowed us to develop Visium HD, which takes the resolution to a single cell scale level. And we have a probe-based chemistry, which is essentially the same chemistry we use with Flex, and so it's human mouse specific. A few months ago or so, we launched a 3' based chemistry, and that is species agnostic, because we capture the poly-A mRNA molecules. This is what essentially gives the single cell resolution scale for Visium. We have essentially a continuous load of oligos across this 6.5 millimeter square capture area. The barcode, the spatial barcode here, changes every 2 micrometer in the X and Y direction. And so that is basically what gives us the single cell scale resolution. And there are no gaps between different barcoded features, so we don't really miss any important information. And the data is really mind-blowing to me, because what you see here is not immunofluorescence, so this is RNA cluster. Different colors represent technically different cell types. So this is a mouse kidney sample, human colon cancer. This was done with the probe-based chemistry. This was done with the 3' based chemistry. And so we can easily profile a mouse xenograft where we have, for instance, human cells injected subcutaneously into a mouse, or a plant tissue. And this week I was in Ann Arbor. I talked to a customer who just ran maize leaves on 3', and again, the data looks fantastic. So this is one of the spatial platform we have. The second platform we have for spatial is Xenium. And if you want to kind of remember what the main differences are between Xenium and Visium, just keep in mind that Visium is whole transcriptome, Xenium is not. So with Xenium, you're going to interrogate hundreds to thousands of transcripts, but you can do that with subcellular resolution. And unlike Visium, where essentially where the output is an NGS-based output, with Xenium, we have an imaging-based technology. So there is no sequencing post-run. That's how much tissue you can profile with Xenium. It's actually, you run 2 slides, so this is 1 slide worth of data. Because it's a targeted approach, we have panels, already pre-designed panels that can be customized with up to 100 custom targets. We have 1 human and 1 mouse panel with up to 5,001 and 5,006 genes. And then we can create fully custom panels with up to 480 genes for pretty much any species, as long as we have a reference we can work with. And we can do also this kind of advanced data analysis application. So if you are interested, for instance, even in express SNPs, we can design probes for that. And with Xenium, we developed a very comprehensive and sophisticated, I should say, cell segmentation methods that uses multiple stainings to segment cells. And so we have 3 different staining for a boundary, for the cell membrane, 3 different staining for the interior of the cell, for the cytoplasm, and then a DAPI staining. And essentially here we have an algorithm that looks at each staining and works in a hierarchical fashion and picks the best combination in order to segment a particular cell type. And again, it works pretty well. I'm going to show you later an image. And finally, Xenium is not disruptive, which means that at the end of the run, you essentially retrieve your tissue. And so you can do an H&E staining, you can do an immunofluorescence staining on the same section, or you can also run CytAssist and essentially generate whole transcriptome data post-Xenium. That's also a possibility. And also we have a panel of proteins. So essentially now you can look at RNA and multiplexed protein from the same tissue section during the same run. And those are the modules that we have. And so essentially to conclude this part, again, I wanted to use this to set up this stage for the demo and start again from this point here where we have this unique toolkit of 3 platforms that now we can use to generate single cell data, spatial data, and in situ data by simply taking consecutive sections of the same blocks, of the same FFP block. This is already been done in a couple of publications, and this is also what I'm going to do during the demo. So before I switch to the demo, do you guys have any questions? All good. So you dissociate the essentially you remove the paraffin, and then you dissociate the tissue and you isolate the CE. So there are yes, there is an enzyme in there. We have 2 protocols, one which is xylene based, and now there is a new xylene free protocol that works pretty well, actually to say. And then I believe we use liberase. I need to double check, but I believe there is liberase in the protocol. And then essentially at the very end of the protocol, you end up with a single nuclease suspension, and then goes into it's nuclear. Yeah, it's because it's FFP, so it's the vast majority of the material we get is nuclear. There are some cells here and there, but it's mainly nuclear. No, so it's a good question. So it's a different technology because with Visium, what we do is we hybridize those probes or we extract the RNA molecules, but the tissue with Visium stays behind. And so we use a CytAssist to transfer either the probes or the mRNA from the tissue onto the gene expression slide. And the tissue at that point is essentially is gone. You won't be able to recover it. With Xenium, it's a little bit different. Essentially, we do a gentle permeabilization, and then we do a probe hybridization, and then we read the signal in situ. So it's really the different way to, and then for Visium, of course, we make a library. So um, yeah, sure. So for the demo, we're gonna work with a human ovarian cancer sample. Now, I'm not an expert in ovarian cancer, and I'm not gonna pretend to be one. So I'm put together some data here, but essentially, we know that ovarian cancer is one of the deadliest gynecological cancer in the US and also across the world. Um, Unfortunately, oftentimes this cancer is detected very late when patients are already in stage 3 and 4. That's why survival ranges between 10 and 40%, so it's really bad. Chemotherapy is a standard treatment, but also we know that there are a good number of therapies now or trials focusing on targeting the vasculature. And so it looks like the angiogenic processes play a really important role in this particular type of cancer, like in many others. And so for this demo, we're gonna focus also on that part. But when we get a data set, essentially there are a few questions that we kind of go through typically, and this could apply to this particular sample. So for instance, we have a tumor sample. We want to understand what the heterogeneity of the sample is. So how many different cell types we have, what different cell types we have. And then can we, in this case, identify these cells that have a angiogenic phenotype? And then once we identify that, can we actually locate those cells across a tissue section? For that, we need to switch to spatial, right? And finally, once we identify those cells, if we identify those cells, and once we identify those cells, can we actually get more information about the niche of those cells? So that is actually how we can use all three platforms together to study the same sample. Generally speaking, we will start with a single cell data, because that is what gives you the heterogeneity. And then we switch to spatial or Visium, and then we switch to Xenium. It's a kind of hierarchical process. So I'm gonna close my presentation here, and I'm gonna switch to Loop Browser, which is, if you guys are not familiar with, so Loop Browser is our visualization tool. It's a desktop app, and so runs on Mac or Windows PC. Super intuitive, and importantly, all software at 10x is free, so you don't pay for anything. And so this is the single cell data. It's about 17,000 cells generated from a FFP block. And so we took a couple of scrolls, dissociate the tissue, get nuclei, and profile nuclei with flex. And this is what we got. So the pipeline identified 20 different clusters, and so here we see the different cluster representation. So each dot represents an individual cell, and cell with a similar phenotype or similar characteristic phenotype clustered together, because they would most likely belong to the same population of cells. But we don't know what cell is what. So we don't know if cluster 4 is a particular cell type. We do have an automated cell annotation tool as part of our cell ranger pipeline, but this data set was not generated using that pipeline. And so we still need to kind of use our knowledge to manually annotate the clusters. Yes. Yes. Yeah. There, yeah, I mean, there could be small differences that, in that case, put one cluster more close to the other one or more similar to the other one. This is just the first pass of the data. Oh, yes. Uh, yeah, I mean, it's probably better reflect the biology rather than having completely separated cluster, which probably is not gonna happen for a real biology. So you might have some cells that are more similar to the green cells, but they are not exactly the green cell in the pipeline. Don't ask me how that works because really, uh, it's not really my background. But it could be similar enough that is put it inside the green dot. Yeah, yeah, but even in the yeah, I mean it does matter where you are. I don't have an answer why this dot is in here. I didn't really look at that, to be honest. But I mean, it would require some more in-depth type of analysis. But there could be those situations where a cell is somehow similar to the green dot and it's put in there, but it's not exactly belonging to the green dot. And the pipeline thinks that, okay, this is actually, it's here instead of being at the edge. But how that is determined, that is really beyond what I know. Yeah. Hmm. Yeah. I mean, and yeah, right. I mean, I would say it's 98% intuitive or 99% intuitive. I mean, you see a green blob here. Those cells are probably different than the orange blob down here. Yes. Okay. Oh, yeah. Yeah. Good point. Yes. Um, but then again, back to your point is, well, we want to know what that little dot or the green blob is. So now we need to somehow annotate those clusters. And there are at least 4 or 5 different ways. I'm not going to show you all of them. But one way is essentially to go here and look at the differential expressed genes in one cluster compared to all other clusters. Since I already done this exercise, I know what is what. And so yeah, I know if I go to cluster number 4, I gonna see cancer-associated fibroblast because I have collagen, multiple collagen genes. I have MMP19, I have DCN. So I can say that cluster 4, that one up there, is probably a cancer-associated fibroblast cluster. Cluster 8, we have PICAM, von Willebrand factors. That's probably an endothelial cell cluster. Right? So that's the way you manually annotate clusters. You can also type your genes or list of genes here one by one and see where those are expressed. For instance, if I type von Willebrand factors, that lights up over there. So this way is not really efficient because you need to enter genes one by one. But what we can do is upload the prepare the gene list where we essentially have multiple markers that are assigned to the specific cell population. And now, for instance, I'm using 3 markers to identify tumor cells, 3 other markers to identify macrophages. Use more markers. I mean, 3 is, to me, is the bare minimum, or 2 or 3 is the bare minimum. That's just an example. But since we are kind of focusing on angiogenesis, I also have 2 markers. I should have prepared 3 on angiogenic cells. And yeah, it looks like they are all pretty much all over the sample, but those are the cells that express either VEGF-A or SRC2A1. If you want to find cells that express both markers, then we switch this combination to feature mean, and now we have a kind of a narrower selection. We can use the lasso tool to basically make a selection here. Oops, make a selection. That's not easy. Make a selection here and say, this is probably my angiogenic cells. So angiogenic cells, I'm going to assign these to a new clusters, for instance, new cluster group. And now I have about 2,700 cells that to me seem to be angiogenic cells. Also, I will encourage you to use machine learning tools to annotate clusters. There are plenty of tools these days. There are a lot of data set, hundreds, if not thousands. And so we can take advantage of those tools and upload a list of machine learning generated clusters that I already prepared. Identify different cell population in here. Now we can see where cells are located relatively better. What is interesting to me is we found, or our analysis found, a VGF-A positive tumor cell, which is down here. Let me actually turn everything off and just turn this on, which if we look at the shape and where those cells are located, they kind of overlap with the annotation I previously made. So that actually gives me two information. One, confidence that the two markers I selected are really identifying angiogenic cells, or at least in part of that. And two, it looks like this angiogenic population is a subpopulation of tumor cells, and perhaps there is really some angiogenic process going on that we want to better understand or better study. Well, the thing is, we are looking at a single cell suspension or single nucleus suspension. Well, and so we no longer have a spatial context. That's why we want to move to Visium, because now we can maintain the spatial context and see where those cells are located. And so I'm going to open a Visium data set. And one thing I actually forgot to mention is the data set I'm going to show you today, so the single cell and Xenium are matched in the sense that we generated the single cell and Xenium data from the same FFP block. This one here is not matched. We didn't have a Visium HD data set from the same FFP block, but we did have a human ovarian cancer sample. So it's not exactly this, it's not the same sample, but it's the same type of tumor. So let's pretend that it's the same. Yes. Yes, for Flex, the vast majority of material that you get at the end of the sample prep is a nucleus suspension. It could be, but there are publications showing that there are a lot of publication on nuclei. For instance, a lot of brain publication are on nuclei because of it's easier to prepare this tissue. They also show that essentially there is no difference in terms of biological information. And one paper I didn't show today is a paper it's a colorectal cancer study. The authors used used FFP blocks to profile nuclei, and they identified and this is published in Cell, they identify a immune exclusion signature that they are thinking about using as a biomarker. And they showed a very nice correlation with survival. So sure, you don't have the full representation, but you do have a very, essentially you look at most of the cases, you look at the true biology of the sample. And I'm from the kidney field. If you look at kidney tissue, or so that it's common to use nuclei because podocytes, for instance, are huge. And so it's much easier to prepare nuclei. But good point, yeah. Now, here, we have, first of all, we have 7 clusters, not 20. It's not the same sample, but again, typically we see less cluster with spatial transcriptomics. Again, we can do the same exercise in terms of cluster annotation. I'm going to just go and focus on cluster number 2 because this is probably, again, a cancer-associated fibroblast cluster. But if we look at cluster number 2, which is this one here in green, that accounts for 30% of the barcode that we detected. So I think there could be more heterogeneity. And so here we can use a functionality in Loop Browser, which is called Reanalyze, to essentially tell the software, search for more heterogeneity. I want to see more diversity in that population because I believe there is something more. I'm not going to run it because it's going to take 5 to 10 minutes, but I already ran the analysis earlier today. And so that's the result of cluster 2 subclustering. So there are 11 additional subcluster. Does it mean that we have 11 additional cell population? Not necessarily, but it means that there are multiple phenotype of those cancer-associated fibroblasts, which is what we will be expecting actually. At this point, I'm gonna go back to my feature here and upload the gene list that I use for single cell. And if we look at the tumor cell, actually, let me change the color scheme here in something that more bluish. If we look at the tumor cell, we actually see a region of dense tumor cell and a region of less dense tumor cell. Why is that? Well, we can, again, use the tools in here to explore those this data. And so once again, I'm gonna use my lasso tool. I'm gonna make a selection here. And I'm gonna call this my dense tumor sample area tumor. And I'm gonna do the same for my less dense. And this, I'm gonna call it less dense. And I'm gonna assign it to my tumor cluster, tumor group. And now what I can do is to ask a loop browser to run a differential expression analysis between the dense and less dense. I want to see what's going on. I want to see if there are biological processes that are maybe enriched. This takes only literally 10, 15 seconds. And what I want to do is focus on the dense region. I want to see if there are biological processes that might be enriched in the dense region. I have my list of differentially expressed genes. What I can do here is export this list as a CSV file. I will export the top 100 differentially expressed genes. And then I'm going to use a tool, this one, gene ontology, to identify enriched biological processes in the dense. And then you can do that the same for the less dense. And so let me just do a quick copy and paste of my list in here. Let's do this, copy. This is very quick, actually, very quick analysis. You launch it. You need to tell it that you're not a robot. Not yet, at least. See the data and sort for raw p-value. And if we look at the enriched processes, there is some terminology associated to angiogenesis. So it looks like there are angiogenic processes enriched in my dense region of the tumor. And sure enough, if I go back here and I look at my angiogenic markers, well, looks like there are more cells in here in the dense compared to the less dense. So that's good. We identified, we have an idea of, or we identify where those angiogenic cells are located across the tissue. But I think we can take this analysis a step further. And so we can ask ourselves what the niche of those angiogenic cells looks like. So what are the other cells surrounding those niches might be looking like? To do that, we need a technology with a high resolution and ideally also a larger tissue section. And so for that, I'm going to switch to Xenium, because with Xenium, we have a larger imageable area and a significantly higher resolution. So let me close the browser, otherwise my computer will get mad at me, and open Xenium Explorer. Now, conceptually, Xenium Explorer is similar to the browser, but it runs on Xenium data. It's free, and what you see here is now we are looking at the matched sample with the single nucleate data that I showed you at the beginning. And what you see here is the tissue that has been stained with our multimodal cocktail for cell segmentation. And so different, we go back here. There are different colors represent the different segmentation staining. You can change you can adjust the parameters here. But if we zoom very close, we can actually see how nice the cells have been stained. This is a way to look at the data. We can look at the data by turning also on the cell tab. And in this case, we have pseudo colors. Let me actually increase a little bit the opacity. And so here, similar to single cell or Visium, different color represent different RNA clusters. So cell with a similar expression profile will belong to the same cluster. And I see that you document. Let me increase it a little bit more. Maybe it's a little bit better. And if we go very close, we can actually see that cells have been segmented based on their shape. So they are not balls, they are not cobblestone thing, but each cell essentially has its own individual shape and its own individual size, its own size. That is what we'll be expecting in biology for a real cell. Since we are talking about cell segmentation, we can also check what cell segmentation method was used to, what staining was used to segment those cells. And then here we can see that the vast majority of the tissue was segmented or cells were segmented using either the boundary staining or the interior staining, pink or green. But there is a pocket of cells down here that have been segmented using the nuclear staining. And why is that? I mean, is that an artifact or is there some biology behind that? Well, we can actually find that out because in Xenium Explorer, we can import pathologist annotation in GeoJSON files. And so I already did that. So let me import my pathologist annotation. And let me actually change the color here. I should have done that earlier, but I want to show this annotation here in white and also this one here in white. And so according to at least our pathologist, those regions here where we see those cells segmented by DAPI staining and nuclear expansion, those regions here are necrotic regions. We can double check that by looking at the H&E staining that is still overlaid on top of this image. If I go back here, actually, let me turn this on, switch to the morphology. And again, we can do that because Xenium is not disruptive. And so post-Xenium, you can run your H&E. And those are your region of necrosis. Now it makes sense that we don't have a good nuclear or a good membrane or interior staining because cells are dying or they are dead, and so they don't have a good staining. And in that case, the algorithm would not just pick any signal, but if the signal is not good enough, it's gonna default back on the DAPI staining. And so, and then if you want to do your own annotation, I'm kind of suspecting that also this region here is another necrotic region. So let's assume that now for a minute I'm a pathologist, which I'm not, but for a second I'm a pathologist. I want to annotate this region so I can select it very quickly like this, make a new selection, assign this selection to the necrosis class, and then again change this color to white and call this necrosis 1. And then you can select more. Again, you can continue with your selection. But remember, we want to find out more about those niches. And so again, we need to go back to a point where we need to identify all different cell clusters. And again, you can look at different markers, but we have a list of annotated clusters that I already have. And so I'm gonna upload a list of pre-annotated cluster. Just give me a second here. Cell. And I'm gonna call this cell types. Okay. All right. And let me open this. And so it looks like we have a lot of those purple cells inside the necrotic region. If we look at here, purple cells represent those are those SOX2-OT positive tumor cells. So now this is very interesting because SOX2-OT is a long non-coding RNA. that is an oncogene and is also becoming a biomarker for different type of cancers. And if I turn everything off and only turn those cells on, now we see that actually it looks like those necrotic regions are really populated by those cells. But what about our angiogenic cells? Here we have VEGF8 positive tumor cell. This is the marker I was using for to identify angiogenic cells. If I turn it on, well, it looks like those cells nicely surround those region of necrosis. And I bet you there's probably here there is another one, and probably here there is another one. And so this basically tells us two types of information. One, now we were able to see what the niche looks like for at least those angiogenic cells. And two, it looks like the necrotic regions somehow are recruiting angiogenic cells. And the other thing I was going to show you with about Xenium is we know that one of the beauty of Xenium is its resolution. And so we can actually see where transcripts are located inside a cell. And so to do that, I'm going to go back to my graph-based clustering. I'm going to zoom very close to cells here. Let me close this one to the cells. And now I'm going to turn on some of the transcripts. So again, we were looking at SOX2-OT positive cells. So here we have the 3 genes that identify SOX2-OT OT positive cells. Turn on the transcript tab. And if you see here, each dot represents an individual transcript, and we can actually see where each transcript is located. So we can easily, for instance, distinguish between cytoplasmic transcript and nuclear transcript. And we can do the same for the markers that identify our angiogenic cells. And so here we have the VEGF8 positive tumor cells. I'm going to turn it on. And again, here, the go a little closer, you can see all those dots that identify those markers. This is one way to see where genes are expressed, but if you want to see, let's say, I want to see only where the all mark or one or multiple markers identifying VEGF8 positive tumor cells are located, is there maybe a quicker way? Yes, there is. Instead of looking at transcript as points, we can look at the data as a density map. If I switch to density map, now I have essentially I have a heat map that shows me where the markers that I selected are expressed across my tissue. And so again, it's a different way to look at your data. And the very last thing I want to show you is again how easy it is to use this tool. So for instance, you create a new selection here, and you want to see what genes are included or are expressed in that particular region and the transcript count. Well, as soon as you make the selection, there is this table that opens up. And here for the genes you activated here, you can see how many transcripts were detected, the density, you can see the cluster the cells belong to, and everything here is exportable as a CSV file. So you can make multiple selection, export these numbers or this data as a CSV file, and then compare 2 or multiple regions essentially side by side. So and because there is no sequencing, the data is available pretty much at the end of the run. And at any point, you can always look at the original web summary file. So let's say we know that those necrotic region are populated by those kind of light blue cells. If you mouse over those cells, Xenium Explorer is going to tell you that those cells belong to cluster 17. And so if you want to know what other genes are differentially expressed in cluster 17, you go here where there is the little I, and then you open the analysis summary. That takes you to the HTML file that is generated by Xenium. And in a second, that's it. There is an analysis tab here. Now, if you scroll down here, there is the log2 fold change table. We can search for the cluster 17. And if we look by p-value, well, actually, first of all, we see SOX2OT is the most differentially expressed genes. And then we can see all other genes that are differentially expressed. And we can already see multiple collagen genes. We see MIH11. So that is not a homogeneous population. It's an heterogeneous population. It's probably cancer-associated fibroblast, smooth muscle cell. That's typical of a tumor microenvironment, having a heterogeneous population. Again, and this is just an example that shows you how you can start an analysis with those tools. But again, the idea, and I'm hoping that I gave you the idea that when you have multiple data set coming from those three different platforms, you can actually look at those in an integrated fashion and start by single cell. Look, that is what gives you essentially the heterogeneity of the sample. Then Visium, it gives you the spatial context, and Xenium really help you dig deeper into the biology, identify those niches. Again, it's a quick example of how you can approach your data analysis. Do not hesitate to reach out to us if you have any question about how you want to approach an analysis. We are here to help you. We have tutorial, FAS is going to be me or Dan or Benton, Eric. So there is a lot of resources. Sandra, take advantage of resources, and we are here to help you. And the last thing was going to put up my PowerPoint again that I closed. But again, I just wanted to reemphasize that at 10x, we have really this unique toolkit of three platforms that can be used as a complementary tools to study the biology of a sample. And with that, I'm going to stop here. I am in this picture. If you find me in the next 30 seconds, I'll buy you a drink. And I'm not the guy in the front that you see there. So this is our CEO. So it's not me, unfortunately. But yeah, thanks for being here. Thanks for your time. And if you have any question, I'll be happy to take any question. Or if there is any question on Zoom, I'll be more than happy to address those questions. Yes. No, I'm not on the front row. I am here. This one here. This, yeah. Mm. Yeah, good. Yeah, so the question is about sample quality. It's a very good question. So because we are profiling RNA, we really want to use high quality samples. So the way we check quality depends also on the type of chemistry we use, the type of application. But let's say if this was the ovarian cancer sample, for FFP, we will be measuring the DV200, which represent the percentage of molecules or RNA molecules longer than 200 nucleotides. That is very strongly recommended for Visium. I will do also for Xenium, and I will also do that for single cell. For Xenium, we would also do a DAPI staining and making sure that the DAPI staining is crisp. Now, we can control how a tissue block is stored most of the time. Sometimes we cannot control how a sample is handled before it being fixed. The recommendation is if you can fix the sample as soon as possible, and do not fix the sample for an extended period of time. So typically it's a 24-hour fixation. Do not fix a 20 grams of tissue because the fixative is not going to get inside. But I mean, again, generally speaking, is an overnight fixation could be 4% formaldehyde or 10% MDF, and then you remove the fixative, you wash it. Once you embed the sample in FFP, that's stable for decades. We generated data. We generated good Xenium data. Actually, we have a customer at the University of Cincinnati who generated really good Xenium data on 30-plus-year-old FFP blocks, and those were pancreatic cancer samples. But that's because the sample at the beginning, fortunately, it was handled really well, and then blocks, as long as they're kept in a dry and cool environment, they last forever, but for a very long time. So, yeah. Yeah, so again, the question is, if I have archived samples, essentially already sectioned on a slide, I would say it depends. The Visium is compatible with that type of slide, the type of samples. We will be still testing the RNA quality. So we have a protocol. Essentially, what we do is we scrape a little bit of tissue and we measure the RNA. The advantage of using this technology is you will have to stain the tissue section before running Visium. And so at that point, if something doesn't really fit, it could be the section has been damaged, RNA quality is too low, you haven't used the reagent yet. So you haven't spent the money yet. You can just discard those samples and maybe prepare or use a different sample, but you are not committed to continuing the workflow. That was the original version of Visium, where sections where the tissue was sectioned on directly on the gene expression side. At that point, you were committed to using. But and again, generally speaking, when it comes to sample prep and sample QC, that also depends on the sample type. I will recommend having a conversation with us. We can bring in the field application scientist, and that is where we can have a in-depth discussion on how to QC your samples, how to prep your samples, again, depending on needs and sample type. Okay. Yeah, yeah. So we generated data, spatial data from bone. The tissue would need to be decalcified. There are different kind of strength of the calcification depending on the type of tissue, how much bone is there. But yeah, it can be done. It can be done.
I also just want to mention, hey everybody, I'm Ben. If you have questions about your specific project, it sounds like this might be a very in-depth sample prep question. Feel free to swing by office hours tomorrow. We'll just be in the atrium here from 9 to 3. So swing by and we can talk about your specific project. Or shoot us an email and we can talk over Zoom.
And the other thing that I forgot to mention is the data set I showed are all available from our website. If you want to take a look at those data sets and you want to continue the analysis, just let us know. I'll send you the links. But they are available. Everything is available.
All right. Well, thank you everyone for attending. So let's give Egon a round of applause.
Thank you.
And as Benton said, if you have any questions, please feel free to reach out to us. Thank you.